Hello and welcome to National 5 Biology, Unit 2, KA5, Transport and Plants. We're still on Unit 2, Multicellular Organisms, and this is Topic 5. Once again, here's the SQA course specification or mandatory knowledge section for this topic. So this is all of the bits of knowledge and content that you can be tested on in a test or exam at this level. As you can see, this is a larger topic, so feel free to pause the video at any point or go back and rewatch sections if you need to. This topic has been broken down into smaller sections to try and help you understand it properly. You can always watch a few parts and then come back again later. As we go through the theory, there'll also be some questions after each part, so you can be sure you're ready to move on. So our learning intention today is to learn about transport in plants, and hopefully by the end of this lesson, you'll be able to state the plant organs and label a leaf structure diagram, describe the parts involved in water and sugar transport, describe the process of transpiration, and describe how the rate of transpiration is affected by various external factors. So the first thing you need to know is that a plant can be divided up into 3 organs: roots, stems, and leaves. If we cut through a leaf to show the cross-section, this is what it would look like if the section was magnified and you would see something like this. So this is the leaf being cut, and if we zoom in on this bit here, this is what it would look like. So you do need to know all of the parts of a plant, be able to label all of the structures and the layers of a leaf structure diagram. So we're going to go through these one by one and test your knowledge. And so for this, what I'm going to do is I'm going to use a simpler diagram. So there are 7 main parts of a leaf that you need to know for National 5. At the top and at the bottom of the leaf are sections we call the epidermis. Epidermis essentially means skin. So there's an upper epidermis at the top of the leaf and there's a lower epidermis at the bottom of the leaf. In the middle, there's the upper epidermis, which is the top, and then there's the lower epidermis, that's the bottom. In the middle are the mesophyll layers, the palisade mesophyll and the spongy mesophyll. The palisade mesophyll, which is this area here, is where the majority of photosynthesis in a leaf takes place. So you can see here that there are lots of tightly packed cells with lots of chloroplasts inside each one. And remember, this is because chloroplasts are the site of photosynthesis. The spongy mesophyll below it gets its name because there's lots of air spaces inside of it, which you can see here. These air spaces are connected to the air on the outside of the leaf by holes in the bottom of the leaf that are called stomata. This means that carbon dioxide can move through the stomata into the air spaces in the spongy mesophyll so that they can be used by the palisade mesophyll in photosynthesis, and oxygen and other things like water vapour can move out of the stomata. So the spongy mesophyll is called spongy because there's lots of air spaces, just like in a real sponge. The stomata, which are the pores on the bottom of the leaf, need to be open and closed at different times. This opening and closing is controlled by special cells on either side called the guard cells because they guard the stomata. So this would be a guard cell here, and this would be another guard cell. And these stomata are all the way along the bottom of the leaf, all of the pores. Finally, you need to know about veins. You can see the veins of a leaf when you look at one when it hasn't been cut open. So you can often see them running along the top or bottom of a leaf that you pick off of a plant or a tree. These veins are made up of 2 really important parts. So the vein is made up of the xylem and the phloem. And we're going to talk about these and their functions in a lot more detail throughout the rest of this lesson. So just to recap one more time, the top section of the leaf is called the upper epidermis, and at the bottom is the lower epidermis. In the middle is the mesophyll layers. The palisade mesophyll on the top is the main layer of cells that are used for photosynthesis in the plant, and the spongy mesophyll below has air spaces to allow gas exchange, and these gases can move in and out through the stomata, and these are pores that are found on the bottom of the leaf. The opening and closing of these stomata are controlled by guard cells on either side of the stomatal opening. Finally, veins run through the middle of the leaf structure, and these veins are made up of 2 vessels, xylem and phloem. So now you should pause the video here and try and label the different parts of the leaf diagram that we've just went through from memory. When you're ready, unpause the video and we'll go through the answers. Okay, so let's run through our different parts. Now, the top part, number 1, you're often going to see in textbooks. This one isn't actually on the mandatory knowledge, so we don't need to know it, but this is actually called the cuticle. So this is the cuticle or waxy cuticle, and I'm just showing you this just in case it comes up in other places. Part 2 is the upper epidermis. Part 3, just below the upper epidermis, this is the layer of packed cells that have loads of chloroplasts, so this is the palisade mesophyll. Layer 4 down here has lots of air spaces, so this must be our spongy mesophyll. And number 5, our layer at the bottom, is our lower epidermis. If we move on to look at 6, 6 is a hole in the bottom of the leaf, so it must be a stomata. And then the guard cells are on either side. So stomata should actually be number 6. Number 7 is the guard cells. So these are just the wrong way around. Number 8 over here is the xylem, and number 9 is the phloem. And together they make the vein. Okay, so what I want you to do now is let's try some quick questions on what we've covered so far to check your knowledge before you move on. So pause the video here and try these questions, either by saying them out loud or writing them down. And when you're ready, play the video and we'll go through the answers. The first question was to name the 3 types of plant organ. So they are the roots, the stem, and the leaves. Then you were to draw and label a cross-section of a leaf, which should have looked something like this. So remember, you have the upper epidermis and the lower epidermis, the palisade mesophyll and the spongy mesophyll in the middle, stomata are the pores on the bottom with guard cells on either side, the vein, which runs through the centre of the leaf, and it's made up of the xylem and the phloem. So all living things, including plants, require water and sugar to stay alive. Plants take up water from the soil through their roots. Plants are also special as they can make their own sugar in their leaves through photosynthesis. Now we'll revisit photosynthesis again in more detail in Unit 3, so don't worry about that just now. So plants synthesize their own sugar and they get water from the soil. But how does the plant ensure all of the cells it contains get access to this water and sugar? Well, the plant has really good transport systems that it can use to allow all of its cells to access the sugar that was made in the leaves and the water that it takes up from the soil. So first we're going to focus on how the plant transports water and minerals as well. So as we go through the details for this part, it's really important that you know all of the structures and processes involved as water moves through the plant, all the way from the soil, all the way to the air at the top. This is quite a common question in the exam, and it could be an extended response question as well. So the first stage of water transport in plants is where water moves from the soil into the plant root hair cells. Root hair cells because they are the cells that form the tiny hairs that are on all the parts of a plant's root. These cells look really similar to the one on the diagram on the right here. Now this topic relies on your knowledge from a previous topic, transport across cell membranes, that was in Unit 1. As you can imagine, there'll probably be a higher water concentration on the outside of the root hair cells in the soil than there is on the inside of the root hair cells. So if there's a higher water concentration on the outside of the cell and a lower water concentration inside the cell, water molecules will move into the root hair cells by osmosis. And there's a reminder of the osmosis definition from Unit 1 on the side here. So that is how water would move into the root hair cells. That's the process osmosis. Now, in the case of minerals, there's a higher concentration on the inside of the cell and a lower concentration on the outside. So if the plant needs to take in minerals, it'll have to be through active transport, as these minerals will be moving against the concentration gradient. So in summary, during the first stage of water transport in plants, water moves from the soil into the root hair cells by osmosis, and minerals move into the root hair cells from the soil by active transport. Once the water and minerals are inside the root hair cells, they then pass into the xylem vessels where they're transported up through the plant to the other cells. This picture on the right-hand side shows what a xylem looks like. You must know the 4 main facts about xylem. So the first fact is that xylem cells are dead. Now the way I like to remember this is that xylem starts with an X, and remember the little dead face emoji that has X's for eyes. So xylem cells are dead, starts with an X, xylem, and you remember dead. Because they are dead cells, the cell walls between the cells are broken down, and that allows the xylem to form a long hollow tube like you can see in this diagram here. The second thing you need to know is that the plants use the xylem to transport water and minerals. Thirdly, you have to know the direction of movement. So the xylem only transports water and minerals in one direction, and that is upwards through the plant. This makes sense as the plants get their water from the soil at the bottom of the plant and they want to move the water up through the plant to all of the cells, so there's no need for the xylem to move water in both directions. It just needs to move it in one direction up the plant. Finally, you have to know about lignin. Now, you can see in the diagram here that there are rings of lignin on the inside of the xylem. So we say that xylem cells are lignified. The reason the xylem has these rings is to help the xylem cells to withstand the pressure changes as water moves up through the plant. If there was no lignin and water was moving up here at really high pressure, then the xylem could collapse. So this is to make sure that it can withstand the pressure changes as water moves through. So to recap, a xylem is how the plants transport water and minerals through the plant. The cells are dead. They transport water and minerals up the plant in one direction. And the xylem contains lignin to help it withstand the pressure changes as water moves through the plant. So once water moves up through the xylem, what happens next? Well, some of the water is used by plant cells for processes such as photosynthesis, but the majority of the water is lost through a process called transpiration. Water leaves the xylem and enters the leaves and then evaporates out of those stomata that we talked about earlier, so those pores that are on the underside of the leaf. You need to know the definition of transpiration. So transpiration is the process of water moving through a plant and its evaporation through the stomata. And you can see that in the diagram here. So water moves in through the plant's roots, up through the xylem, and then evaporates out of the stomata and the leaves, and this process is called transpiration. So let's try some quick questions on what we've covered so far, just to check your understanding. So pause the video here, try these questions either by saying them out loud or writing them down, and when you're ready, play the video and we'll go through the answers. Okay. So for question number 1, you were to name the vessel in a plant that carries water and minerals. The answer for that is xylem. For question number 2, are the cells that make up this vessel dead or alive? They're dead. Question 3 was, in what direction or directions does this vessel transport water and minerals? It's upwards or in one direction. Question 4 was, why are these cells lignified? And it's to withstand the pressure changes as water moves through the plant. Finally, we asked, what is transpiration? And it's the process of water moving through a plant and its evaporation through the stomata. So the next area you need to know about is the rate of transpiration. The rate of transpiration is how fast transpiration happens. So how quickly does water move through the plant and evaporate through the stomata? The rate of transpiration can be affected by abiotic factors such as wind speed, humidity, and temperature, and it's these 3 you need to know for the exam. So these factors, wind speed, humidity, and temperature, can either increase or decrease the rate of transpiration. One key thing to remember is that light intensity has no effect on the rate of transpiration. The light intensity changing on a plant has no effect on how quickly the water moves through the plant and evaporates through the stomata. So we're going to look at these one at a time. So firstly, if we consider wind speed, if the wind speed is increasing, so the wind is moving faster across the outside of the leaves, water will evaporate out of the stomata quicker as the water vapour is being moved away from the outside of the leaf quicker. So increasing the wind speed increases the rate of transpiration. Secondly, if we consider humidity, we need to know what humidity is first. So humidity is the concentration of water vapour that is present in the air. So if there's an increased humidity, so a lot of water vapour present in the outside of the stomata or the leaf, then this will slow down the rate of transpiration as water vapour will be slower to leave the leaf. So this is the only factor where if you increase it, the transpiration rate will decrease. However, be careful, because if they ask a question about what happens if there's a decreased humidity, so the air outside of the stomata will be drier to water vapour will be able to leave the stomata at a quicker rate, so transpiration would increase. So for humidity, always remember it's the opposite. If the humidity increases, the transpiration rate will decrease, and vice versa. The final factor is temperature. So if temperature is increased, then they'll increase the transpiration rate. So it increases as the water will be quicker to evaporate through the stomata because the temperature is higher. Now, how would we measure transpiration rate? So we can use a piece of apparatus that is called a potometer. And this is one of the key pieces of equipment that you could be asked about in your National 5 exam. Potometers are used to measure the rate of transpiration in plants, and there are two versions of potometer which often come up, and we're going to look at both of these individually. So the first one is this one here, which is a bubble potometer. Now you can see the setup of this potometer in the diagram. You place a plant or a leafy shoot with the roots in the water at this end, and then at the other end you have glass tube running through, and then you have a beaker of water. The water can be either coloured, or you can introduce a bubble inside the glass tube so that you can see how far it's travelled, so you can measure it and measure the rate. If water is lost by transpiration through the plant shoot, so the water is moving up through the plant and evaporating through the stomata, then basically water will then be pulled up through the glass tubing into the roots to replace that water. So the upwards movement of water through the glass tubing can be measured as how far the coloured liquid or the bubble passes. And we can easily measure this either using a ruler or using a scale that goes along here. However, because this is rate of transpiration, just knowing the distance isn't enough. We have to take account of the time. So what you do is you could measure the distance it's travelled, the bubble or the coloured water, and then you divide it by the time it took, and that will give you the rate of transpiration. So if the bubble or coloured liquid moves quicker in the time given, then the transpiration rate would be faster. Another way to set up the potometer, instead of measuring distance of coloured water or bubbles, is to measure weight or mass. So what you do with this one is you take a plant and you place it in a container of water on top of a balance. You measure the mass at the start of the experiment and the mass again at the end of the experiment. Now this works based on the fact that the plant loses mass as more water is lost through transpiration through the stomata. So if water is taken up from here, goes through the plant and leaves, there'll be less water in here, which means the mass will be lower. Once the start and end mass have been calculated, the difference can be divided by the time taken again so that we can get the rate. A final thing that they could ask you about this setup is why there's a layer of oil on the surface of the water. Now the reason for this is if there's no layer of oil, the water could evaporate into the air straight from the container and not by moving through the plant. And this could result in a loss of mass that wasn't caused by the water moving through the plant, so it wasn't caused by transpiration. So it would mean we'd be measuring the wrong thing. So having a layer of oil prevents the water from evaporating straight into the air and means that any water that is lost has to have gone through the plant through transpiration. A common question that's asked in exams or tests is if you were the experimenter, how would you alter the apparatus to measure the effect of external factors on the rate of transpiration? So things such as wind speed, humidity and temperature, which we talked about earlier. So if you wanted to take the potometer apparatus that we just looked at and measure wind speed, what you could do is just add a fan and you could change the speed setting to simulate wind, and you could see how fast transpiration was at higher wind speeds or lower wind speeds. If you wanted to investigate humidity, you could basically place a plastic bag over the plant shoot, and the buildup of water leaving the plant and being trapped in the bag would increase the humidity or the concentration of water vapour in the air that was surrounding the plant. Finally, if you wanted to measure temperature, you could just use a heater to change the temperature beside the plant. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. So for the first question, it asks you to name the piece of equipment which can be used to measure the rate of transpiration. Potometer. For question 2, what is the effect of an increase in wind speed on the rate of transpiration? When it asks you for the effect, they just want increase, decrease, or stay the same. So it would increase it. For number 3, what is the effect of a decrease in humidity on the rate of transpiration? Well, remember, humidities are one that's the opposite. So a decrease in humidity would mean an increase in transpiration rate. And number 4 is asking, apart from wind speed and humidity, name one other factor which will affect the rate of transpiration. We know that must be temperature. So now we've looked at how plants transport water and minerals. Now we're going to move on to learning about how plants transport sugar. So we mentioned earlier how plants synthesize sugar in their leaves through the process of photosynthesis, and that they must have a system to get this sugar to other cells within the plant. So plants transport sugar through something called the phloem. So this is the other part of the vein that we talked about earlier. Like the xylem we talked about earlier, you have to know 4 main facts about phloem. Firstly, unlike the xylem cells, phloem cells are living. They're used to transport sugar, and unlike the xylem cells, they transport sugar in both directions, so up and down the plant. Finally, you have to know that phloem cells have sieve plates, and they also have associated companion cells. You do not need to know what these do, you just need to know they're present inside the phloem. So let's try some quick questions on what we've covered about phloem and check our knowledge before we move on. So pause the video again here, try these questions by saying them out loud or writing them down, and when you're ready, play the video and we'll run through the answers. So for question number 1, it said to name the vessel in a plant that carries sugar, that is the phloem. Are the cells that make up this vessel living or dead? They are living. In which direction or directions does the vessel transport sugar? It is up and down the plant or in both directions. Name the 2 parts that make up this vessel are the sieve plates and the companion cells. So one of the most common questions you can get asked in an exam is to compare the xylem and phloem vessels. This could potentially be an extended response question. So to help with this, I've created a summary slide that shows you the 4 facts about each so that you can compare them directly. So firstly, you can compare whether the cells are dead or alive. So to do this, you could say xylem cells are dead, whereas phloem cells are living. So remember, you want to do an actual comparison, you want to put the word whereas in between. Next, you could compare the substances that they transport. So you could say that xylem transports water and minerals, whereas the phloem transports sugar. Next, you could compare the direction of transport. So you could say that the xylem transports water and minerals in one direction, upwards in the plant, whereas the phloem transports sugar up and down the plant in both directions. Finally, you could compare the vessels and structures. So you could say xylem cells are lignified to withstand the pressure changes as water moves through the plant, whereas phloem cells do not contain lignin but contain sieve plates and associated companion cells. So I hope that this comparison will make those questions easier. So that's us finished about learning about transport in plants. I hope that you can now state the plant organs and label a leaf structure diagram, that you can describe the parts of the plant involved in water and sugar transport, and describe the processes of transpiration. You should also be able to describe how the rate of transpiration is affected by the 3 factors that we discussed earlier. Please feel free to go back and watch parts of this video again in future if you need a refresher on the topic, but thank you once again for listening.